Can you tell us what the Olympus trial found and just a little bit about it and summarize it for us?
For sure, yeah. So for those who may not be familiar, the Olympus trial was a study looking at a formulation of mitomycin mixed with a unique hydrogel. The hydrogel has unique properties in that when you cool it down, it's a viscous liquid. It does have very finicky chemical properties, so there's only certain things that can be admixed in it. One of them turns out to be mitomycin, which they can put at fairly high concentrations, dissolve it in there. And then when the gel goes up to body temperature, it becomes like the consistency of Jello, essentially. It essentially becomes a slow release formulation of mitomycin. And it dissolves in urine at a fairly constant rate. And so when that drug was developed, and the company was looking to use it for this disease, what we started doing was getting compassionate use approval through the FDA. So for every single patient, we had to go through the FDA to get compassionate use approval. And this was after animal safety studies and everything were available. And by doing that on a, not even, I don't think it was more than a dozen patients, basically provided some background data to show its efficacy. Based on that, they developed this Olympus trial, which was using this formulation. It was single arm, so not even comparison, because the FDA accepted the fact that there was no standard available, right, other than hysterectomy, and their very strong inclination was, its potential ability for kidney preservation. And so the bar was set very low in terms of what the standard had to be met. And I can't remember exactly if it was 18% or 25% response rate. And then there was no defined limit on adverse events because they were willing to look at what the risk benefit was. So I have to say, this was my first significant interaction with the FDA. I ended up going there a couple of times to talk.
Cool.
And I remember going there saying, well, they're going to want my opinion on stuff and I'm going to school them on upper tract cancer. And 15 people walk in from the FDA, ranging from like statisticians to PhDs. I was so impressed with their level of knowledge, for this rare disease that we think have a total, complete handle on, at least, whatever we think we know. I was really impressed. I mean, it was humbling in the level of knowledge they had broadly and deeply, So, I learned a lot from that interaction. And so, clearly a lot of their effort is safety. But one thing that it was very interesting to me that, you think they're going to be so rigid about parameters, and they weren't. They said, well, we accept the fact that we don't know what the response is going to be, and we don't know what the adverse event rate is going to be. So let's just look at that when you have the data.
Wow.
If the responses are really good and maybe the adverse events are not great either, that's still okay.
Right. We don't know.
Out. So basically, to get back to answering your question, what this study did was that we took patients with low-grade upper tract cancer, biopsy-proven low-grade. Tumors had to be 1.5 centimeters or smaller. They could be multiple tumors, but all of them had to be that size or smaller, so you could laser them ahead of time. If they had ureteral tumors, they had to be laser bladed. There could not be active ureteral disease.
Sure.
And then basically after that, they were eligible to receive the drug once a week for 6 weeks. Now, there was no prescription as to how it would be delivered. It could have been done by nephrostomy tube or retrograde, but I think all of us were nervous about doing it through a nephrostomy tube. We'd never done anything like this. And so everybody did it retrograde for the trial. And so once a week for 6 weeks, and then 6 weeks later, they had to have a ureteroscopy, and a complete response had to be seen. So in other words, we had residual index tumors that we then treated. So it wasn't just adjuvant, it's, a new paradigm for us, right? Chemoablation, like literally ablating residual tumors. And basically what the study showed, I think it was 79 or 80 patients total. There was a 59% complete response rate. And then with the follow-up data that we have, in those who did have a complete response, there was roughly another 56% that had durability over the course of a year. And so that trial helped establish this as a new standard for low-grade, low-volume upper tract cancer. We did have a high adverse event profile, so roughly 35–40% rate of ureteral narrowing or stenosis. I certainly saw that a lot in my practice. I think we learned a lot during the trial. The mistake that I made, and I think others made, was that we noticed the narrowing. We didn't know what to do with it, but since they were on a trial, we kept going. And that was a mistake. What I think we've learned since then is that partly it appears to be related to the repeat mechanical trauma, probably combined with the caustic effects of mitomycin. And what you initially see is edema, probably, the narrow edema. And we think it's maybe similar to what you see in the bladder, which could be an eosinophilic type reaction. And then what we've learned is that if you just leave it alone, it usually goes away. And so the thing to do is, if that happens, you put them on a drug holiday. I mean, it is low-grade disease, right? It's not really that necessary to be super aggressive. So we leave it alone, put them on a drug holiday for 2–3 weeks. We treated with steroids, based on very low evidence, but knowing that it could be inflammatory.
Right.
And so that seems to mitigate the concern for development of permanent strictures. And the ones that develop permanent strictures, they were awful, permanent strictures, and that needed definitive management later. So I think those are the lessons we've learned. And then since the trial has completed, people have been exploring other ways of doing it.
Do you think a percutaneous or an antegrade approach may be helpful in minimizing stricture risk?
I think there's definitely promise there. I was very reluctant to do it. I was a little bit worried, but there are other leaders in the field who actually took that initiative to start doing it.
Right.
Kate Murray, especially, and Jen Linehan, who were involved with the trial as well. And so I had heard that they were doing it, so I called both of them up to talk to them. And they had done a few patients here and there without an issue. They just made, they did diligence. They made sure that the tube went in a uninvolved calyx, so that there was no tumor in the calyx that it went through. Did antegrade fluoroscopy before treatment to make sure that the tube was in good place, stuff like that, right?
Right, sure. So
I basically followed their lead and mimicked, their process. And I can tell you now, and I'm told I'm the highest volume user in the United States because I get so many of these patients. So now, past couple of years, this is the only way I do it. And we definitely do not see the strictures or the narrowing or anything. I get I integrate fluoroscopy on every patient still, but I think I'm going to stop doing it. It hasn't, as long as they're not having trouble with the tubes and there's no hematuria, I'm not sure we need to be doing that. And much easier to do, of course they have to live with a nephrostomy tube for about 7 weeks or so.
Sure.
But we do once a week treatments through it. Once that we know how much volume to put in, you do have to assess the volume of the renal pelvis to know how much volume of drug to give. And so it's much easier. It's actually easier for them, except for the fact that they have to have a nephrostomy for a little bit. But, patients get used to having a port, for chemo one way or another. And so this is just another concept there. And so, as soon as that treatment's over, we take it out. I let everything heal up, and then I scope them 6 or 8 weeks later. And I have to say, it's been much easier on my practice because I do have a lot of these patients. It's a very quick office visit as opposed to a procedure.
Sure. It must be better for the patients too, right? Like
we think so. What we don't know is it equal efficacy or not? And then the safety, I think we need more solid data, and it is starting to come out. There is one publication out there. It's retrospective, of course, but there should be more coming out. And look, there's also no real science to having to do it once a week for 6 weeks, especially for my more frail patients or who have to have a family member take off work to bring them in. I easily tell them not to stress about the fact that if they have to come every 2 or 3 weeks to do it, generally for these low-grade tumors, they don't have a high proliferative rate. So I think it's, it's okay to, we don't have data, but there's no reason to think that you're shortchanging patients by modifying the protocol a little bit.